Okay, so today I'm going to be doing mycology lectures, 2 mycology lectures, and I'm not going to take a break between the two because both of them are like 55 slides. So I should get done with this in probably an hour, hour and a half, and then so you can be released early. And then tomorrow, hopefully we can do some we'll probably do a test in the laboratory. I'm not quite sure. Listeria. So I have listeria growing. That'll be an interesting, fun lab. Remember, listeria has the umbrella motility, and we'll try to demonstrate tumbling motility. And then we'll do some tests. Hopefully, the camp test will work, or hopefully, I'm sure the bioesculin will work for listeria. But anyway, that's for lab tomorrow. And so we're gonna have a short lab tomorrow, and then we'll have the review for Friday's exam. So same format. I'm gonna do the review. I'm gonna do the review twice. It seems like when I do the second review, you guys have already taken in the information, and which is good. And I think you guys have become better prepared for the exam on Friday. So I think what we're doing is working out. Okay, are there any questions at this point? Does anybody know what's going on next Thursday and Friday, April 28 and April 29? I got an email from Deanna saying that she wanted to know if I was going to either start late or release you guys early. So I don't know if she wanted to borrow you, or I don't know. She wanted to know what's going on with the class. If not, I can shoot her an email and find out what's going on. All right, let's go again.
She wanted to have the externship meeting, but we're still trying to figure out which is a good date for everybody.
Oh, really? The externship as far as your clinical rotations?
Yeah, she wants to have a meeting prior to class time.
So is this for like next Thursday or Friday?
We're not sure yet, because she had initially asked us for Friday, but it turns out not everyone is free before class on Friday. So she sent an email trying to see when we're free basically in the next 2 weeks, and then decide from there based on everyone's feedback. So we don't have a date yet.
Okay, because I'm sure, because one of the options was either before class, if I was going to start late, and I've never started late, but or the other option would be to leave early, and you guys have been leaving pretty much early because of either probably no lab. Like, for example, well, we are going to have a little bit of a lab tomorrow, but no lecture, so you guys will be leaving early tomorrow. All right, stand by for information. Hopefully Deanna will get back to us. All right, mycology. Okay, so what I've done was I have two mycology lectures. Okay, so when I took mycology in my pre-internship years, mycology was a subject all into itself. So I had 10 weeks of medical mycology. So what I've done was I'm going to be giving you the basics of mycology, and then towards the end of the lecture, I'm going to get into the clinically significant fungi. Okay, so objectives, you'll be able to describe and identify, describe safety precautions, lab methods, and identifications. Okay, definitions. Make sure you know the definitions, because those every time you have definitions, or the definitions that you have on your homework, those are good matching questions. Okay. So mycology, study of mycology is the branch of botany dealing with fungi. Comes from the Greek word mycos, which means fungi. Mycosis, anything ending in osis, means it's some kind of disease. So mycosis is a fungal disease, a disease caused by fungi, a fungal infection in or on a part of the body. Fungi, a kingdom of plant-like spore-forming organisms. The only difference even though it's described as plant-like, there's no chlorophyll, so there's no photosynthesis. So fungi grow in masses without roots, stems, leaves, or photosynthetic pigments. Okay, no chlorophyll. Sporangium, another term, is a large sac-like structure, a closed sac-like structure where sporangiospores are formed. Sporangiospores are what propagates the organism so it can continue to grow. The spores are released and they spread, and then they make more fungi. Eukaryotic, it's a single cell or multicellular organism whose cells contain a distinct membrane-bound nucleus, and that's the identifying characteristics of a eukaryote. It has a membrane-bound nucleus. Okay. Blastocanidia Blastospores. These are new terms, and they refer to the spores of fungi. Like, for example, I'm going to talk about Aspergillus. I'm going to talk about Mucor or Ascidius or Rhizopus. Those are fungi that have Blastocanidia or Blastospores. So these spores, the Canidia or Blastocanidia, are spores formed by budding, as in a yeast. Okay, they're thin-walled and water balloon-like. So they look like if you can imagine looking at a daisy, at the end of the petals of a daisy, then all these little circular buds form. So that's what a fungus would look like, and I'll show you pictures of it. The little yeast-like canidia, those small cells, are called canidia, and those are actually the spores. It's kind of like, if you can think about if you go back, if you go back to hematology, remember the nurse cell in the bone marrow, and the outermost edges of that nurse cell, or the megakaryocyte. Remember the megakaryocyte? At the edge of the megakaryocyte, what happens? Well, what happens is that's where the platelets slough off. Okay, but in a fungus like Aspergillus, the canidia form at the end, and what happens, those canidia are actually spores, and they get released. And like I said, when they get released, it propagates the life of the organism by developing in another location. So those are the spores or canidia. Another type of canidia is chlamydocanidia or chlamydospore. I just talked about blastocanidia or blastospores. These are spores formed by the rounding up of a cell, thick-walled, intercalary, or terminal position, and it's not shed. Okay, so these are not released. Heterotrophic is an organism that cannot make its own food. Okay, so since it cannot make its own food, then it has to get it elsewhere. Okay, like us, we don't make our own food to survive. No, we have to go to the store or wherever for our nutrition. So it's an organism that cannot synthesize its own food, such as an organism that's saprophytic. Saprophytic is a special type of plant that derives its nourishment from dead or decaying organic matter. Okay, so a saprophyte lives off of dead material. Septate and aseptate. Septate is hyphae. So if you think about a tube, that's what hyphae are. Hyphae are tubes, or they're branches, okay, that are subdivided into individual cells by transverse cell. So if you think about a tube that's divided into sections, for example, bamboo. I'm sure you've all seen bamboo, and bamboo has along the stalk of the bamboo plant are walls that separate out the bamboo plant into sections. That's what septate means. Okay, so septate hyphae are hyphae that are. What happens is that there are segments in that hyphae that are compartmentalized, just like bamboo. Just imagine bamboo, right? Aseptate means without walls. So you can imagine bamboo or a straw, for example. If you look at a straw, there are no walls compartmentalizing areas of that straw. So that's aseptate. And what septate is if you can imagine bamboo, because the segments of a bamboo plant has those walls. Okay, so hopefully those two analogies will help you out. Septate versus aseptate. Hyphae, like I mentioned, basic structure units of mold, tube-like projections. In these tubes, like I said, hyphae can either be septate, which has those walls, or aseptate. Pseudohyphae are elongated buds. Okay, so usually yeast, like for example, Candida, will multiply by budding. So what happens is one area of the bud will grow and it'll grow and then it'll just pinch off. That's budding yeast. Okay, but pseudohyphae, the buds have failed to separate, meaning it has failed to release. And as a result, that bud is sausage, looks like the link of a sausage. All right, and that's pseudohyphae. Mycelium is basically, and I'm sure you've seen this in, like for example, in a culture plate. You've seen a culture plate where what's growing on that culture plate are the fuzzies. Okay, if you see the fuzzy, the fuzzy fungus is what you're actually seeing are mycelium, and they're the aerial. It's called the aerial mycelium because what you're actually seeing is the fungus portion that is not connected to the media. So if it's not connected to the media, that's called aerial mycelium. Remember, mycelia or mycelium are the fuzzies, the fuzzy part of the fungus. Okay. On the other hand, so the aerial part is away from the media that it's growing on, and then I'll illustrate this later. And then as opposed to what's called the vegetative mycelia. The vegetative mycelia is what's connected to where it's growing from, like the auger media or the substrate. Okay, the substrate. So there's two types of mycelia: aerial and vegetative. Like so, vegetative is nutrients absorbing and water exchanging portion. The water exchanging portion means it's getting water from the substrate beneath it. Okay, so the vegetative is on the bottom. Aerial is above the vegetative. It extends above the substrate. Okay, actually, hopefully you got my explanation. Aerial extends above the substrate. Well, vegetative extends above the substrate, but it's still attached to the substrate. Aerial is above the vegetative mycelium. Okay, there's an illustration later on. Yeast is a unicellular, is one cell fungus that reproduces by budding. Okay, and a good example is Candida or Candida albicans. We have some in the laboratory, and we'll play with it a little bit tomorrow. Okay, so well, you see how you got these compartments here with nuclei. This is septated hyphae, septate hyphae, septate. Like I said, compartments with its own specific unique nucleus. Okay, septum. Here, there's no septate. There's a cell wall, but there's no sectioning off of the compartments. So this is aseptate or non-septated hyphae. The correct term is aseptate. Same nucleus, but no walls. Okay, remember these small balls here are Canidia, So these, on this particular fungus, it's called Cryptococcus. Well, and on this particular fungus, it's Blastocanidia. Okay, so here you got aerial reproductive hyphae, then aerial vegetative hyphae. This is vegetative because it's connected to the substrate. And then you have penetrative hyphae, where it has penetrated below the surface of the substrate, and those are basically the roots. So penetrative hyphae are the roots. Okay, aerial vegetative means it's above substrate, but it's still connected to the it's above the substrate, but still connected to the substrate. And then the aerial is above the vegetative hyphae. That's the aerial hyphae. Okay, precautions. Whenever you handle funguses, fungi, you need to be working in a laboratory that's either BSL 2 or BSL 3. And you need to be in a hood class 2 or class 3. Okay, you don't want to handle those funguses because of the spore generation. And if you accidentally, God forbid, inhale those spores, you can get pretty bad lung infection, okay, respiratory or lung infection. So avoid aerosols. That's why you need to work underneath the hood. Eukaryotic protists possess these are just characteristics: nucleus, nuclear membrane, endoplasmic reticulum. So that means there's protein synthesis going on. So you know the endoplasmic reticulum, there's two types, right? Smooth ER and a rough ER. The smooth ER is the endoplasmic reticulum without ribosomes, and rough endoplasmic reticulum, it's rough because it has the ribosomes still attached to it. And as you know, ribosomes are involved with protein synthesis. Then you have your Golgi apparatus, mitochondria for energy, and a rigid cell wall. So that's a eukaryotic protist. These, like I said, are single forms. They're easy to see on the gram stain. If you look on a gram stain, or if you look in, if you look on a gram stain, you'll see, look for your gram-positive cocci, chains, pairs, clusters, right? Pairs, chains, and clusters. But then all of a sudden, you see this large black egg-shaped organism. That's a yeast. It's large, it's black, and it's egg-shaped. That's a yeast. So I'm going to have you take a look at under the microscope what yeast look like on the gram stain. Okay, there are multiple cells that form filaments. Those are molds, unicellular, multicellular, and fungi lack chlorophyll. They're heterotrophic, can't make its own food. Saprophytic, remember saprophytes survive off of dead decaying material, and or parasitic. So they'll attach to an organism and steal nutrients and parasitize a host. Many are ubiquitous. Ubiquitous means it's found it's all around, and they're reproduced by spores. Okay, I showed you the chlamydospores and the Chlamydocanidia and the Blastocanidia. So those canidia are actually the spores, and they may be derived directly from the mycelium. Remember, mycelia are the fuzzies. Okay, if you see anything on a plate, and sometimes the fuzzies are so numerous that it may actually push off the lid of the petri dish. So that's why you need to be handling fungi underneath the hood. Hypha or hyphae. Okay, so you should be able to be forming an idea, getting an idea of what hyphae look like. They're the tubes, tubular filament or threads, branched or unbranched. Septate and aseptate hyphae, compartmentalized or not compartmentalized. Mycelia. Okay, so you get the feeling that this lecture is being repetitive. So mycelia, again, massive hyphae, vegetative and aerial hyphae. Okay. True hyphae or filamentous flat-ended cells from transitional cells. They do not show the points of constriction. And characteristics of pseudohyphae. Remember, pseudohyphae are hyphae that have started to bud and elongate, but they have not pinched off. So they end up looking like sausages. Regular points of constriction, linked sausages produced in nutritionally poor environment, and they may bud to form blastospores or yeast cells. 3 types of clinically significant hyphae: choanocytic, that means it's sparsely septate, that means the cell walls are there, but they're not close to each other. They're located far apart. And then you got pigmented hyphae, septate of the Mediatious fungi. Okay, that's pronounced Mediatious. It's a specific type of fungus, and I'm going to get into that later on. A Mediatious, to determine if you have a Mediatious fungi, you'll take your plate and you just turn it over, and if it's dark or black, that's a Mediatious fungus. That means it's pigmented, as opposed to the septate hyphae, which are usually non-pigmented. You turn over the plate and you look at the media, and there's no dark brown or black. Okay, so the Mediatious hyphae are the pigmented hyphae, and they're usually there's also septate. And then you also have septate non-pigmented hyphae. So two types of septated hyphae, pigmented and non-pigmented. Molds, you've all seen molds. Molds are cottony, woolly, and they're woolly. Why? Because they've got mycelia. They're cottony. Why? Because they've got mycelia. Those are the mycelia. Powdery or fluffy. Optimum temperature for growth of molds is room temperature. Okay, that's why molds are all over the place. In dark room temperature areas. Yeast, on the other hand, what they look like on culture are smooth, pasty, mucoid, butyraceous. Butyraceous means having a butter consistency. Okay, butyraceous is buttery consistency. Mucoid, you know what mucoid is? Pasty. Okay, what pasty means, to describe it, it's like it has kind of like a dull luster. It's not shiny. If you look at, for example, a colony of Staph aureus, if you look at it real closely, it's a white colony, and it doesn't have a luster, but it's kind of shiny. However, on a yeast are also white, but they have a dull kind of like pasty color. It's a dull white. Hopefully we can see isolated colonies of yeast tomorrow, and you'll know what I mean. So if you look really closely at your white colonies when you do when you're doing a throat culture or any kind of culture, you see a white colony, don't always assume that it's going to be some kind of staph or some kind of strep. Depending on, because both staph and strep, the colonies are kind of shiny. They will, if you look really closely, you'll see that shine. Yeast, on the other hand, will have a pasty look. It's dull, and it kind of has that dull kind of luster. Okay, pasty looking. The yeast grow optimally at 35, 37 degrees. Okay, so those have to be incubated. Dimorphic fungi. Okay, what a dimorphic fungi are. I've talked about molds, which are the mycelium, the branches with the hyphae. That's one type. That's actually one morphology. The other morphology is the yeast form. Some fungi have dimorphic morphology. They can either have a yeast form or they can have a mold form. And usually the medically important fungi are dimorphic. For example, H. capsulatum is Histoplasma Capsulatum. That's a medically important fungus. A Blastomyces dermatitidis is another important fungus. Penicillium marneffii, Coccidioides immitis. I'll talk about these later, and Precautions brasiliensis, and Sporothrix. I'll be talking about this towards the end of the second lecture. But these are the medically important fungi. They happen to be dimorphic, okay, meaning that they have a yeast form and they have a mold form. The mold form is when it has the branching hyphae, and the yeast form has only the unicellular portion. Okay, only the unicellular portion is present. Mold mycelial saprophytic phase produce delicate hyphae. The mold form of colonies have cobweb or hair-like appearance. Okay, it has the fungus, the fuzzy form, or the mycelial phase. The yeast phase, yeast tissue parasitic phase, can grow on media with cyclohexamide or some kind of antifungal agent. Usually, the media on fungal cultures have a lot of antibiotics. Why do they have antibiotics? Because when doing a fungal culture, you don't want staph or gram-negative rods. You're not interested in the staph, gram-negative rods, or whatever. All you're interested in is there's the fungus present. Okay, that's why the media for isolating fungi contain antibiotics. We don't want the interfering bacteria. This is mycology. Bacteria is in probably on another culture. Okay, so here, oops. So here you can see the different types of forms for dimorphic fungi. On this half of the page, on the right side, are the yeast forms, and then on the left side of this column are the mycelial forms. Okay, this is these are all pathogenic or medically important funguses like Blastomyces dermatitis, B-derm, Coccidioides immitis, which causes valley fever, and I'll talk about these later on in the second lecture. Histoplasma capsulatum, that's a medically important fungus, and that's important for, then you get that from bird droppings, either bats in the caves or from starlings, for example, or pigeons. So you get that from bird feces. Coccidioides brasiliensis, Candida albicans is a pretty common yeast. We have Candida albicans in the laboratory, and we'll be doing a test to identify Candida albicans, and that's the germ tube, this one right here. You won't see germ tube in nature. You have to create conditions where a germ tube will form. And if the germ tube does form, then you can call it Candida albicans. Cryptococcus Neoformance. The big deal about Cryptococcus Neoformance is that this outer circle here is actually the edge of the lipid capsule. Cryptococcus Neoformance is a capsulated yeast. So the way you actually see it, and there's going to be a picture later on in the lecture, is that you use what's called an India ink prep. So you make a suspension of your Cryptococcus, and we'll do this in the laboratory, and you make your suspension with Cryptococcus, and then underneath the cover slip, what you do is you put just a little bit of India ink, and it's black. You don't want to put too much or everything will be black. You only put so much that you can actually see the capsule of Cryptococcus Neoformans repel the India ink particles. Okay, when it repels the India ink particles, what you have are the yeast just lights up your field of vision underneath the microscope, and they look like headlights. Okay, so that's Cryptococcus Neoformans. You'll actually see the capsule repel the India ink particles, and hopefully we'll see this in the laboratory. And we were able to see it last semester. And then finally, Aspergillus. Aspergillus is a common fungus. There's a yeast form here, and there's with the Canidia here. That's what Aspergillus looks like. Okay, pigmentation. Demediaceous is dark. Hyaline means no color. So demediaceous means it's pigmented, and hyaline means there's no pigmentation. Growth rate is significant. Culture media. So when we do the cultures, we keep my fungus cultures for 4 weeks. Okay, AFB cultures are for 8 weeks. So if we do AFB fungal cultures in the middle of the AFB cultures, we're at the point where we're throwing away the fungal cultures, because fungal cultures are only 4 weeks. Okay, media contain either blood, chocolate, etc. Those normal can be normal tubes. A battery of recommendations, with or without blood, with or without cyclohexamide, or with antibacterial agents. Almost all of them have antibacterial agents. There's large culture tubes for fungal cultures. They're poured in thick slants. If you remember what our slants look like in the laboratory, they're thick. They're these big thick tubes and they're slanted, and then you just streak the organism on the surface, and you look for growth, no growth. Okay, and always remember that when you are incubating tubes, you never screw it down tight. Advantage of the large culture tubes: it's easily stored, less space, doesn't take up too much space, easily handled. You just lift up the tube, look for growth, no growth, and less dehydration, because you don't have much of a surface area, but that could be a disadvantage. So you got poor disadvantage of the large culture tubes is poor isolation, and like I said, reduced surface area for growth. Petri dishes is another way to do fungal cultures. And the advantage is that it provides larger surface area for growth. Mixed culture is easier to separate, so you can handle your fungal growth easier with the plates as opposed to the tubes, because you have less of a surface to be dealing with it. And it provides maximum aeration. Disadvantages of the Petri dishes: the Petri dishes have a tendency to dehydrate and dry out, and hazardous culturing of certain systemic mycosis would include Histoplasma, Blastomyces, Coccidioides. You have to use these underneath the hood, especially when you're dealing with Histoplasma, Blastomyces, or any of the medically important fungi. BHI Auger BHI stands for brain heart infusion, with or without antibiotics. Other media include DTI, which is dermatophyte test medium, or DTM, inhibitory mold auger, mycophil auger, cornmeal auger. Cornmeal auger is media that's used to isolate Candida albicans. And then there's Microcell auger and Sabrode with brain heart infusion. Antibiotics of the chloramphenicol and cyclohexamide, those are used to minimize growth of bacteria because they interfere with the growth of the funguses. For the diamorphic fungi, you got your mold fungi that grow at room temperature, and your yeast will grow at 35 to 37. BHI auger is also good for the recovery of saprobic and pathogenic fungi. So BHI augers is another routine media that you can use for fungal isolation. Sabhi is another form of brain heart infusion, and it's good for the isolation of significant fungi from mixed flora specimen like sputum. Whenever you do a sputum culture, I don't know, maybe we should probably do it where you guys do throat swabs and then you streak it onto media, and might want to see what grows. Microcell auger fungus is selection for used to isolate funguses. It's also for the isolation from specimen with normal flora. Again, it helps inhibit normal flora, and it's good for dermatophytes. The dermatophytes are fungi that infect the surface of your skin, the dermis, okay? Those are the dermatophytes. DTM, dermatophyte test medium, selected for dermatophytes again. Infectious and dermatophytes are those fungi that infect the skin, hair, and nails, dermatophytes, dermis. DTM is used for screening purposes only, and then once you get growth, then you subculture it to medium without antibiotics. Cornmeal auger, like I mentioned, is good for Candida, and so we have Candida albicans growing in the laboratory. So I don't know if we have cornmeal auger, but it seems to grow pretty well on chocolate agar. So we'll play with Candida in the laboratory. Chapex agar is for the differential identification of Aspergillus species. Actually, all of them will grow Aspergillus. Just don't think that Chapex is only for Aspergillus. So it's just another type of media that will grow fungi. Specimen processing and identification. Laboratory safety and mycology safety is always important in the microbiology laboratory. It's no different here for mycology. So all mold and cultures must be handled in a type 2 hood, class 2 biological safety cabinet. You also too want to keep pathogenic isolates sealed. The gram stain, rule of thumb, all fungi stain gram positive. Spore and hyphae stain gram positive. Yeast stain gram positive. If we deal with the candida, if we are playing with the candida tomorrow, I want you guys to make a gram stain so you can see what candida looks like underneath the microscope. Candida albicans, as all of the fungi, appear black. Large black eggs, okay, oval shape. KOH, potassium hydroxide. The KOH prep is used for like skin scrapings or nail scrapings where there's a lot of keratin involved. The KOH, what it is used for is to hydrolyze or dissolve the keratin. And you use 40%, so it's relatively high concentration. It's for skin, hair, nails, and other tissue. Okay, so if you're dealing with skin, hair, nails, and other tissue, you want to get rid of the keratin. So this is after a KOH prep, and what's happened is the KOH has dissolved all the keratin, leaving behind these fungal elements. You see, what you're seeing is our hyphae, yeast, and my fungal elements. So you can report this as fungal elements noted, hyphae and yeast forms. Same here. This is skin scraping with a KOH prep. The KOH dissolved the keratin, and this is what is left over on the KOH prep. Thrush is a type of disease. It's caused by Candida albicans, and thrush is if you have poor oral hygiene and you have a Candida infection, that Candida infection is called thrush. Okay, cutaneous mycosis. Okay, so now we're going to talk about the different types of mycoses. The different types of fungal diseases. So these types of mycoses are cutaneous, and so they infect the surface of your skin. For the Woods black lamp, that's basically an ultraviolet light to detect infected hairs by fluorescing a bright yellow-green lamp on the head of the patient. And if you see the green fluorescence, that means it's a positive Woods lamp. So what you would do is you would recommend the patient wash the affected skin sites with 70% isopropyl alcohol. Okay, so for these cutaneous mycoses, you take small fragments, and these are scrapings. They take a scalpel blade and scratch, and get skin scrapings or nail scrapings. And if you receive tissue and you want to do a KOH prep, then you can add KOH, but you have to mince the meat with the blade, chop it up a little bit, and then add your KOH. And then hopefully you'll be able to see the fungal elements. And then so when you add your KOH to the skin scraping, then you cover it with a cover slip and then read microscopically. If the keratin is not dissolving, you can accelerate the dissolving by heating it underneath the same way you did the Zell-Nielsen stain with the lit applicator stick. Then once you applied heat and the KOH dissolved the keratin, then you press your cover slip down. Of course, make sure it's cool, and then look for the fungi microscopically. Okay, clearing apparent as change in preparation to turbid. So initially it'll be clear and then it'll change to turbid. Thick opaque samples are unsatisfactory for examination. That's why you have to chop it up and use the K-Wedge to dissolve that excess tissue. And thick samples will also obscure hyphae and spores. So you don't want thick samples. That's why you have to chop up the sample. Interpretation, hyphae demonstrate uniformity in size and symmetry. If it's there, you'll definitely see it. You see branching hyphae, you see yeast, okay? But that your reporting protocol will have to depend on your specific laboratory's SOP. Positive for dermatophyte, well, that may be one laboratory's way of reporting the presence of a dermatophyte, but usually laboratories will report fungal, no fungal elements seen. So it's like NFE, that's usually the code, no fungal elements seen. Or if you do see fungal elements, then you'll either say few mycelia, few hyphae, or few yeast forms noted, okay? So you report the presence of mycelia, the presence of hyphae, branching hyphae. I don't know if you can tell septate versus aseptate, probably not. You'll just see the hyphae. And then also too, if you see yeast, then you report yeast noted. You can't just say positive for dermatophyte. Okay, tinea. Tinea is the name of the fungal disease. The tinea is a type of mycosis, okay? Tinea versicolor is short, stubby hyphal elements and grape-like clusters of spores. Like tinea pedis is athlete's foot. Tinea is, like I said, it's the mycosis. It's the fungal infection. Positive for Candida, you see pseudohyphae, chlamydospores, and blastospores. If you see mosaics, they are not fungi. Those are cholesterol deposits or cell wall skeletons. Hyphae do not grow geometrically. So if you see something that looks like a nice geometric or prism-like pattern, that's probably not a fungus. It's probably some kind of artifact or cell wall skeleton. Okay, that's the end of Mycology 1. So I'm going to move right on to Mycology 2. Okay, lactophenol cotton blue. Lactophenol cotton blue is a stain. It's a nice, pretty blue stain, but it's a background stain for fungus preps, and you're able to see the chlamydospores really nicely after you do a tease mount or any kind of preparation. You counter stain it with lactophenol cotton blue, and you should be able to see the like I said, the fungal elements really nicely. So again, when you're dealing with or working with fungi, you need to work underneath the hood, biological safety cabinet 2 or 3. Take care special care in obtaining inoculum with a needle. Now, when you do that, when you're dealing with these mold with a needle, that's called getting a tease mount, because you're actually teasing the fungal colony, and you're taking a chunk of that fungal colony and you're making a prep, okay? But when you're doing that, when you disrupt the chlamydospores or blastospores, for example, you need to be working very, very carefully, and you also need to be working underneath the hood.
Professor?
Yes.
The next PowerPoint is not showing.
Oh, okay. You know why? Because I didn't share it. I'm sorry. New share. How about now?
It's good now. Thank you.
Okay, so going back to lactophenol cotton blue, like I said, it's a nice, pretty blue stain. It's kind of like a counter stain, so you can see the fungal elements really nicely, work under the hood, etc. Okay. All your preps need to be underneath the hood for safety reasons. Add a few drops of stain, and that should be enough. Gently heat it so you can see the elements. That's optional. And you look for the mycelium, the arrangement of the mycelium and the fruiting structures. The fruiting structures, like I said, are the canidia. Like here, these are lactophenol cotton blue preps. See how the nice. Okay, so this is why I didn't like mycology too much, because my preps like this, these are like aspergillus. This could be aspergillus. Were never this nice. All I got were balls. All I saw were the canidia, but I didn't see the main structures. Okay, when I did my T-SMAs. But these are lactophenol cotton blue preps. Okay. The reagents, negative staining. Negative staining is, remember I talked about Cryptococcus and the headlights. Cryptococcus has a capsule, and that capsule actually repels. It repels the India ink particles. It repels the India ink particles such that the capsule on the microscope looks like it's they look like headlights. Okay, we did the negative staining. We do have Cryptococcus in the laboratory, and we do have India ink. So we'll be doing an India ink stain with Cryptococcus, and hopefully we'll be able to see the negative staining of Cryptococcus. Okay. Yeah, it uses India ink, and it's good for identifying Cryptococcus Neoformance, which is a pretty significant medically important yeast. It's a yeast, and I actually did see that in a real situation one time where I saw it was like towards the end of my shift. And usually they'll do a request for India ink on spinal fluid, because this type of yeast, the Cryptococcus Neoformans, has a tendency to affect the nervous system of patients. And so what they'll do is, if you do a spinal tap and it's purulent, of course they'll order a regular bacterial culture, but they'll also order India ink. A lot of CSFs, or if not most CSFs, they'll order India ink. And it was towards the end of my shift. I was supposed to get out at 3:30. At 3:25, I get the CSF with the India ink prep, so I do it real fast. And I made my prep, added India ink, and guess what I saw? I saw a whole bunch of headlights. So I knew what I was seeing, so I reported it to the ER. I didn't have to get the pathologist to confirm. As soon as I saw those bright headlights, then I knew that it was Cryptococcus. Like that. This is what a positive India ink prep looks like. You see the brightness right there? That brightness is because the capsule is repelling or resisting the India ink particles. Okay, so hopefully we'll be able to see that in the laboratory. Like I said, we do have Cryptococcus, and we do have India ink. Negative staining. Candida albicans. Candida albicans is a yeast, so on the gram stain, they look like large black eggs. Okay, we'll see that on the gram stain. So that's just typical yeast, right? But the doctor wants to know if the patient has Candida albicans, because Candida albicans is pathogenic. It'll cause disease. So we'll do the germ tube test, and the way you do it is you prepare a suspension of the Candida albicans yeast. Okay, you make a suspension, and what you make the suspension with is human serum. So today we'll do this test next week. So what we'll do is tomorrow you'll draw serum on each other or somebody, and then all we need are 0.5 ml aliquots. So if we can get nine aliquots of 0.5 ml serum, maybe that's one or two students. All we need are serum aliquots. Daniela will freeze them, and then next week we'll do the germ tube test. The germ tube is not seen in nature. You have to force the formation of germ tubes, okay? So the germ tubes are, like I said, you make your suspension using human serum. What you do is, once you add the yeast to the serum, then you incubate it at 37 degrees for 2 hours, okay? And at the end of 2 hours, you'll see the germ tubes. And these are tubes, they're not hyphae. These are actual tubes that come off of the yeast. And if they come off at 90 degrees, or they shoot out at 90 degrees, they're not really too long, but you'll see it looks like a little straw coming out of the yeast. That's a germ tube. And at the end of 2 hours, you look at it microscopically, and it's either Candida albicans if you see the germ tubes, or yeast not Candida albicans. Okay, so this is a good test to identify the presence of Candida albicans in your specimen. So 0.5 ml human serum, inoculate the serum for 2 hours at 37. So you'll inoculate your serum, and we'll do other things like probably the review. At the end of the review, then you take a look at the specimen underneath the microscope and look for germ tubes. Okay, they're hyphal extensions, but they are not buds. Okay, they're not pseudohyphae. So make sure pseudohyphae means at the end of the hyphae you'll see pinching. Germ tubes look like straws just coming out of the yeast cell. Okay, if it looks like sausage where you see the constriction, that's pseudohyphae. That's not a germ tube. So 90-degree angle looks like a straw coming out of the yeast cell. Yeast assimilation test. Okay, so this could be a board exam question on how do yeast handle carbohydrates. Well, they don't ferment. Excuse me. Yeast do not ferment carbohydrates. They do not oxidize carbohydrates. What they do is they assimilate carbohydrates. Okay, yeast assimilate carbohydrates, or carbon and nitrogen sources. So if positive on the yeast assimilation test, you'll show growth or turbidity, considered along with morphology and reproductive structures. There's one system called the Uni-Yeast Tech, where you can identify Candida albicans on that wheel, but you can also identify another species called Candida tropicalis. And the way I can tell immediately Candida tropicalis is that Candida tropicalis is positive for trihalosis. So TR goes with TR. Candida tropicalis is another non-pathogenic form of Candida, but it's tropicalis. And that particular yeast is positive for trihalosis. The API 20C is for the Vitek yeast card and the yeast tech microplate. Okay, though these are other commercial kits used to identify yeast. Yeast fermentation test consists of an organism suspension plus carbohydrate. If you see gas bubbles, it's rarely used. Like I said, yeast do not normally ferment carbohydrates. They assimilate carbohydrates. Okay. And it's replaced with the assimilation test, and it's used as a backup test. Slide technique, slide culture technique. Okay, this is, I don't know if we'll have time to use this because fungi take a long time to grow, but the way the slide techniques are done is that you use fungal media like Sabdex Auger, okay, Sabdex Auger, and you take a little square out of the auger, you cut it out, and you put it on the on a slide. okay? And then you take, you do a little tease mount, get your culture, and put it on the on your chunk of agar that you placed on the slide, and then you cover slip it, and then you incubate that. And what you're doing is you're letting the fungus grow underneath the cover slip. Okay, so cut out a 1 centimeter block square, inoculate each corner of the block, place heated cover slip directly on the surface of the agar block. Okay, then you incubate it until you incubate you put the slide with the agar block on the cover slip, incubated at 30 degrees centigrade until you see obvious growth. Once you see obvious growth, then what you'll do is you'll lift the cover slip, because the fungus has grown underneath the cover slip. You take the cover slip off, and then you put it on a glass slide, and you add lactophenol cotton blue, and you examine microscopically. So the fungus, it's growing on the agar, the Sabdex agar, but it's also growing on the cover slip as well. So when you see the growth on the cover slip, then you take that cover slip, place it on another slide, add lactophenol cotton blue, and then examine microscopically. So that's a slide culture technique where you're using a chunk of agar, just like this. Okay, chunk of agar goes onto the slide. There's your fungus culture. So looking at this picture, there's a couple things wrong, right? Okay, you got that fungal culture. That's pretty dangerous the way it's sitting out like that. So when it's exposed like that, you better be underneath the hood. And because now you're exposing all that fungus to the air, and you're potentially breathing it. So that better be under the hood. The other thing is the guy's not using any gloves. So you got the chunk of agar there. You take a piece of it using a tease mount, put it on that agar block, place a cover slip on top of that, and then you incubate it at 30 degrees and look for growth after you're incubating it. Tease mount. I hate the tease mount. Tease mount is when you take a needle. You've seen the needles, and then you just go into the fungal culture, take a chunk out, and carefully put it on the slide, and then add lactophenol cotton blue to see the fungal elements. Okay, using a dissecting needle or pointed applicator stick, you dig out a small portion of the colony to be examined, place it on the slide, add lactophenol cotton blue, and examine microscopically. Okay, you can examine it 10x, 40x, and under oil immersion. And this is why I don't like the tease mount, because teasing colony disrupts delicate fruiting structures. Well, guaranteed, when I did a tease mount, I definitely disrupted fruiting structures. And all I saw were Canidia. And that's why I didn't like mycology too much. The Scotch tape preparation, again, if you go to your culture like this, what you'll do is you'll take a piece of Scotch tape, sticky side down, and you touch the sticky side onto the culture, and what you'll do is you'll tape that Scotch tape onto a glass slide, place lactophenol cotton blue underneath it, and then you can see the fungal elements. This I like better than the tease mount, because you're using the tape to get the natural form of the fungus. So you're not disrupting anything. What you're doing is you're putting a piece of sticky tape on top of the colony, and you're pulling it off and taking it and putting it on a glass slide, so you can examine it microscopically after you add lactophenol cotton blue. Okay, it's good for the diagnosis of tinea versicolor. Tinea versicolor is a rash caused by fungus. Tinea is a type of mycosis. So tinea is a type of mycosis. Okay, so you take a piece of tape, portion of that rash with the sticky side down, pull it off, put it on a slide, add lactophenol cotton blue, and look for fungal elements. It's kind of like a gentle form of skin scraping, except you're taking off the part of your skin with the sticky side of the tape, and then you're making a taping it to the microscope slide, and then you're looking at it microscopically. Okay, other mycological examinations, periodic acid shift, where it's what that does, that stains periodic acid shift. It will stain fungus is black, fungi black. If you see the black, then that's a good indication that fungus is present. And also to nocardia. Nocardia is, remember that it's partially acid fast, but it's also stains. It doesn't stain well with periodic acid shift. But just reserve nocardia for the partially acid fast portion on your test. Calcofluor white stain used to detect fungi. It's used with KOH, and KOH is a clearing agent, and the calcofluor will show fluorescence. So the calcofluor is a fluorescent examination. Okay, the difficult part is that you need to have a fluorescent microscope, and not too many labs have a fluorescent microscope. So you do your KOH, and then you add the calcofluor. The calcofluor will show fluorescence if funguses are there. It's kind of like the Woods lamp, right? The Woods lamp is ultraviolet light. If it's there, then that portion of your skin, usually it's the head, tinea capitis, will have a fluorescence, green fluorescence. So this one is a similar principle, but it's using a culture. Okay, mycoses. This is the last section. This is actually the material that's testable because these are fungal diseases. The first one are the cutaneous mycoses. The cutaneous mycoses. So cutaneous meaning on the upper layer of the skin. So that means these are probably these fungal diseases are caused by dermatophytes. Remember, dermatophyte, dermis, okay? Superficial scaling. If you have itch, won't go away, it's probably some kind of fungal infection. So you might have hyphae there, and then you might want to do that Scotch tape test. Take the Scotch tape, place it on where it's itchy, take it off and look at it microscopically, and hopefully you won't see any hyphae or fungal elements. Clinical characterization, habitat from geophilic or zoophilic, meaning from the ground or animals. Clinical types designated by Latin binomial tinea. Okay, tinea or tinea capitis or tinea pedis means infection of the head, or tinea capitis, or tinea pedis. The other name for tinea pedis is athlete's foot. So it's a fungal infection of the foot, fungal infection of the head. That's tinea capitis and tinea pedis. Microsporum. Okay, Microsporum is the genus. There's only one organism on Microsporum, and that's Microsporum odwini. Okay, that's pronounced odwini. It's the most important cause of tinea capitis. So make sure you know that. So now I'm telling you, make sure you know this. A lot of the organisms in this section of the lecture, I will tell you that make sure you know them. Okay, tinea capitis in school children, so adolescents. It's spread by direct contact with infected hairs on the caps, haps, combs, and clippers. Causes hair to fluoresce underneath Woods lamp. Okay, remember Woods lamp. So that's Microsporum odwini, most common cause of tinea capitis in school children. Microsporum Canis causes inflammatory tinea capitis, same thing. Zoophilic, so it comes from animals, usually acquired from puppies or kittens. That's Microsporum Canis. Trichophyton. So don't confuse the T for trichophyton with the T for tinea. So Trichophyton mentagrophytes. Trichophyton mentagrophytes. That's the most common species of trichophyton, okay? Remember that, Trichophyton mentagrophytes. And the infection, it causes tinea barbae, which means the beard, tinea capitis, which is the head, tinea corporis, which is fungal infection of the trunk of your body, Tinea pedis, which is the foot, and onychomycosis, which are your nails, okay? Your nails. Trichophyton rubrum, Trichophyton rubrum, T. rubrum, is the second most common form of trichophyton. Remember, the most common is Mentagrophytes. The next one is rubrum. And rubrum causes pedis, porporus, cruris. Cruris is like jock itch, and onychomycosis, which are the nails. And then Trichophyton tonsurans causes capitis, pedis, porporus, and onychomycosis. I'm not going to ask you, because a lot of these are common. You don't have to memorize that all the different types of tinnias under each trichophyton, okay? Like those are the different tinnias under Mentagrophytes. Those are the different tinnias under rubrum. And those are the different tinnias, capitis, pedis, etc., under tonsurans. So when I took medical mycology, I had to memorize that. So when you're CLS level, you probably might want to create a table to say which trichophyton will cause what tinea disease. Malassezia furfur is another fungus that causes tinea versicolor. Microsporum gypsum, another fungus, causes corporus capitis, and it's recovered from hair and skin. This is another important one. Remember the name, Epidermophyton flocosum. It's like Microsporum odwini. This is the only organism in that genus. Epidermophyton flocosum will cause trinia cruris and tinea pedis. Okay, so you got the jock itch, and then you have the athlete's foot. Okay, a lot of athletes have this. They scratch one area and they actually infect the other area. That's what happens. You got jock itch and then athlete's foot. That's caused by Epidermophyton flocosum. And then you have Candida albicans, one of the most common yeasts. Candida albicans is a yeast, causes cutaneous candidiasis. If you see the disease monoliasis, monoliasis is an infection caused by Candida albicans. Make sure you know that monoliasis is caused by Candida albicans. Systemic diagnosis in immunocompromised individuals. So blood cultures, if it's positive for yeast, it's chances are Candida albicans. And from the mouth, it causes thrush. Okay, Candida albicans infection in your mouth is the disease is called thrush. That's the oral infection of Candida albicans. All right, pictures, more pictures. Okay, now those were the cutaneous mycosis. Now we got the subcutaneous mycosis. Now we're going deeper in. Clinical significance caused by fungi inhabiting the soil, decaying vegetation, usually induced by trauma. That means if you have a wound or if you run into something that has fungus on it, then that's how it's introduced into your body. You got disseminated forms. Some individuals are predisposed to systemic infections, like the immunocompromised people. All right, make sure you know this one. Subcutaneous mycosis, Sporothrix. This one is, you'll see this on your test for sure if you get any mycology questions. Sporothrix causes what's called rose gardener's disease. So what'll happen is you'll get a case study of a gardener who came down with infection, and where he accidentally pricked his finger on a rose stem. You know the thorn of a rose. Well, unfortunately, that contained the fungus Sporothrix. So his wound never healed. He had an antibiotic. He added medicine to it. It never healed. As it turns out, they did a fungal culture, and it grew Sporothrix. So Sporothrix causes what's called rose gardener's disease. Okay, rose gardener's disease. You get it from getting a finger prick, like I said, when handling roses or bushes. And the fungus that grows is Sporothrix schenki. Make sure you remember that one. Okay, looks like this guy had a fight with a whole bunch of roses and he lost. That's Sporothrix schenki. Sporothrix schenki, and looks like the Incredible Hulk right there. Significant infection. Okay, and then you've got the deep mycoses. Okay, that was the subcutaneous. Now we're getting into the deep mycoses. Deep mycoses, clinical significance, soil fungi usually involved. Infections due to inhalation of spores, because now you're inhaling the spores deep into your system. Disseminated forms invade the organs. Each species has a favorite organ. Certain individuals are predisposed. When I talk about predisposed, a lot of these predisposed individuals are like the immunocompromised. Those are usually predisposed. Coccidioides immitis. Make sure you know this one. This one causes valley fever. Coccidioides immitis, and it's common. Well, it's common, like remember in Francisella. Francisella causes tularemia. Well, that was in Tulare County, California. Well, around in that same geographical area, the San Joaquin Valley, that's kind of like near Tulare County. Unfortunately, that causes another disease called valley fever, where the farmers would be having respiratory problems because the deep mycosis, you get infected by inhaling the spores. Particularly in that part of California, the fungus is Coccidioides immitis. Unfortunately, if you're positive or if you inhale the fungus Coccidioides immitis, is really, really hard to get rid of. Okay, it's really, really hard to get rid of. So you inhale the spores, you'll try to treat it. What they'll do is they'll probably do what's called cocci serology for Coccidioides immitis, and usually your serology is positive for this kind of fungus. The next one is Histoplasma Capsulatum. Histoplasma Capsulatum is a fungus you get it from inhaling the spores that come from bird droppings, like either bats, and your disease that you have, you contract, it's called Spelunker's disease. What a spelunker is a person who explores caves. Okay, so, and in these caves are the bats. So if you inhale the spores that come from the feces of bird droppings, then that's chances are it's Histoplasma capsulatum. But it's not only bats. You can get it from starlings, usually in the Midwest, where they have a lot of starlings, swarms of starlings or pigeons. It's, again, the fungus is, you get it from the bird droppings of these birds. Okay, bat, pigeon, it's the bird droppings that contain the fungus. So that's Histoplasma capsulatum. You get Spelunker's disease for bird droppings, or even pigeon droppings. No particular disease. It's called just histoplasmosis. Okay, histoplasmosis or Spelunker's disease. Histoplasma capsulatum, bird droppings. B. dermatitidis is another deep mycosis. We won't be identifying that, but I know that this type of yeast form has what's called broad-based budding. Paracoccidioides brasiliensis is another fungus. Pictures. Okay, opportunistic mycosis. Clinical significance, non-pathogenic fungus that causes subcutaneous and disseminated infection. Disseminated infection means it gets into your system and disseminates throughout your body. In immunosuppressed or debilitated patients like HIV patients, treatment with corticosteroids, cytotoxic drugs, and antimicrobials. Patients with diabetes mellitus, okay? These individuals, HIV and diabetic patients, are prone to getting these fungal infections. Diabetes, because high sugar content, yeast love sugar. Okay? They will assimilate all that sugar. and HIV because they are immunosuppressed, they are prone to infection. So a lot of diabetic, because their sugar count is high, yeast will go after that. Zygomycosis, zygomycetes, and the main ones are Rhizopus, Mucor, and Absidia. I'm not going to emphasize too much on these zygomycetes because these three, Rhizopus, Mucor, and Absidia, are all over the place. They are, you put a plate out, if bacteria grow, that's expected, but if fungi grow, chances are it's going to be Rhizopus, Mucor, or Absidia. And then there's Cryptococcus as an opportunistic mycosis. Cryptococcus, like I said, is a clinically significant yeast, okay, because it can infect the central nervous system, and that's when it's detected on a CSF sample. Like I said, think about the time when it was like almost closing time for me. I had a CSF that I had to read. I did in India ink prep, and guess what? I saw a bunch of headlights. So that was Cryptococcus Neoformance. It's not a good fungal infection to have because, like I said, it infects the central, the cerebrospinal fluid, causes your CSF to be purulent. And then Aspergillus causes aspergillosis. Aspergillus, make sure you know this, is the most frequently encountered fungus in the lab. Okay, I think I'm going to make that red, but it's the most frequently encountered fungus in the lab. That's Aspergillus. Aspergillus is also all over the place. It's the most common species. Penicillium marnefii, from mucocutaneous infection to disseminated infection. So you can get in your upper respiratory, and then it can become disseminated throughout your body. That's Penicillium marnefii. All right, pictures. There's a negative stain, India ink stain right there. And I think that might be like Aspergillus. And that's a chunk of lung infected. All right, are there any questions? That's it for my mycology. 2 lectures combined into one, and that's mycology, and that'll be your next exam after this coming Friday. So this shouldn't be a long test. 2 lectures, 2 homework to do Monday. All right, any questions? Okay, so if not, we'll do a little bit of lab, and then we'll do the review for your Friday's test tomorrow. Okay. So if there aren't any questions, have a good evening, and I'll see you tomorrow.